How can affinity to oxygen change? Well, first of all, it must be a protein with multiple binding sites, and those binding sites must be able to interact with each other through a phenomenon called cooperativity. With positive cooperativity, the first binding event increases affinity at the remaining sites. You can also have negative cooperativity where the first binding actually reduces affinity at the remaining sites. So here is a picture of what that would look like. So first you have no ligand. Much of the molecule is either flexible or somewhat unstable. Then you have ligand binding to one of the subunits. This binding stabilizes a high affinity conformation, and more of the structure is stable and none is unstable. Then the rest of the polypeptide takes up a higher affinity conformation, and this is stabilized in the other subunit through protein-protein interactions. And then a second ligand molecule binds, and that binding occurs with a higher affinity than the binding of the first one. And this is an example of a form of induced fit. We can quantitatively describe cooperative proteins using something called the Hill equation. So if we think about a protein with multiple ligand binding sites, we would describe that with this equation. P plus NL, where N is the number of ligands that can bind to the protein, gives you PL sub N. And then we take the concentration of the products over the reactants to get to this Ka expression. Now when we then rewrite that in terms of theta, we get to this description. Now we can take the log of both sides, which gives us the Hill equation, which is in the form of an equation for a line, y equals mx plus b. So here this would be y, this would be b, this would be x, and then the n value would be the slope, which is called the Hill coefficient. So if n is equal to 1, there is no cooperativity. If it's greater than 1, there's positive cooperativity, and if it's less than 1, there's negative cooperativity. So this shows you the Hill plot of myoglobin and hemoglobin. So for myoglobin across the range of pressures of oxygen, there's no cooperativity, but for hemoglobin, in the high affinity state, there is a cooperativity where NH equals 3. Cooperativity is a special case of allosteric regulation. An allosteric protein is one in which the binding of a ligand to one site affects the binding at a different site, and it can be positive or negative. If it's homotropic, then the normal ligand of the protein is the allosteric regulator. If it's heterotropic, then a different ligand affects the binding of the normal ligand. So cooperativity equals positive homotropic regulation. Now let's examine more the R and T states of hemoglobin. So T stands for 10th state. In this state there are more interactions and it's more stable, and there's a lower affinity for oxygen. R is the relaxed state where there are fewer interactions, it's more flexible, and there's a higher affinity for oxygen. When oxygen binds, it triggers a conformational change from the T state to the R state. And this involves breaking ion pairs between the alpha 1 and beta 2 interface. So this is a close-up view of a portion of hemoglobin in the T state without any oxygen bound. So the interactions between the ion pairs histidine HC3 and aspartate FG1 of the beta subunit are shown, and then also between lysine C5 of the alpha subunit and the same histidine HC3 of the beta subunit are shown with the dashed lines. Now pH also has an effect on the ability of oxygen to bind to hemoglobin. So here we have 3 different curves at 3 different pHs. So are you noticing here, does acidity increase or decrease the Kd? So again, to find the Kd for each curve, we look at the 0.5 value on each curve. So at a pH 7.6, that Kd to me looks like it's, I don't know, maybe 2.7. Then at the curve for pH 7.4, that Kd looks like it's closer to 4. Let's call it 3.8. And then if we look at the curve at 7.2, that Kd looks like it's above 4, maybe 4.3 or so. So as the pH decreases, the KD is increasing. What that means is that as the pH decreases, as acidity increases, the affinity for oxygen is lessening. Actively metabolizing tissues generate H+, which lowers the pH of the blood, which then would decrease the affinity of hemoglobin for oxygen. So in those cases, that's where hemoglobin would be releasing oxygen. The hemoglobin affinity for oxygen depends on the pH because H+ will bind to it and stabilize the T state, that's the low affinity state. And it does so by protonating a particular histidine residue known as histidine 146, which then forms a salt bridge and leads to the release of oxygen in the tissues. And the pH difference between the lungs and the metabolic tissues increases the efficiency of the transport of oxygen. So that is just further emphasizing the ability of hemoglobin to release oxygen in the tissues and to bind it in the lungs. And the effect of H+ and CO2 on the binding and release of oxygen by hemoglobin is called the Bohr effect, which is named after Christian Bohr, a Danish physiologist who first discovered it in 1904. Now, CO2 is also produced by metabolism in the tissues and must be exported. And in fact, 15 to 20% of CO2 is exported in the form of a carbamate on the amino terminal residues of each of the polypeptide subunits in hemoglobin. And the formation of a carbamate yields a proton that then further contributes to the Bohr effect. And the carbamate additionally can form salt bridges, further stabilizing the T state. So what I'm saying here is that CO2 and H+ both will favor the T state and decrease the affinity of hemoglobin for oxygen. Now, there is one other factor to consider in how hemoglobin binds oxygen, and that's this molecule 2,3-bisphosphoglycerate shown here. So this is not a normal ligand, so this would be a heterotropic regulator, and it's present at millimolar concentrations in red blood cells, and it's produced from an intermediate in glycolysis. It's a small negatively charged molecule that binds to the positively charged central cavity of hemoglobin and stabilizes the T state. So this is a picture of it binding to the central cavity in the T state. It will not bind in the R state. Now, this molecule allows for oxygen release in the tissues and adaptations to changes in altitude. Now, in normal human blood, the concentration of BPG is about 5 millimolar. But that's at sea level. At high altitudes, it increases to be about 8 millimolar. The reason it increases at high altitudes is because at high altitudes, oxygen delivery declines by about 1/4 to 30% of the maximum. Therefore, an increase in BPG concentration decreases the affinity of hemoglobin for oxygen, allowing it to be delivered to the tissues at the percentage that is normally delivered at sea level. The final topic is sickle cell anemia, which is due to a mutation in hemoglobin. In sickle cell, a glutamate residue is switched to a valine residue in the beta chain of hemoglobin. And the new valine side chain can bind to a different hemoglobin molecule to form a strand similar to the amyloid proteins that we discussed in Chapter 4. This sickles the red blood cells. Untreated homozygous individuals generally die in childhood, but heterozygous individuals exhibit a resistance to malaria and experience much less severe symptoms. And it is thought that the evolutionary adaptation for this is that it does give an advantage to people for resisting malaria, and if you're just heterozygous, then you can still live with the disease as long as you avoid chronic stress or excessive exercise. And this movie shows how that one exchange of the glutamate for the valine results in these strands that can form. So there's the valine, and now it's going to interact with a second subunit. So now that's formed an octamer, so it's two of these hemoglobin tetramers, and this can continue to bind to other tetramers via additional valine hydrophobic pocket interactions, which then results in strands that deform the red blood cell, giving it a sickle-like appearance. In summary, the affinity of a protein for a ligand is typically expressed in terms of the equilibrium dissociation constant. The lower the Kd, the greater the affinity of the protein for its ligand. The Kd for a protein can be determined by graphing the fraction of bound sites indicated by a capital Y or a theta versus the concentration of the free ligand. Both hemoglobin and myoglobin bind oxygen, but only hemoglobin binds oxygen in a cooperative fashion, where it switches between a T state and an R state. The T state is the low affinity state, the R state is the high affinity state. It can also bind carbon dioxide and H plus at different sites, and it binds those with an inverse affinity compared to oxygen. And when CO2 and H plus bind hemoglobin, that stabilizes the T state and encourages release of oxygen. So that's going to happen in the tissues. And then there's also the BPG, which can regulate the affinity of hemoglobin for oxygen as well.